Hello guys, welcome back to our channel mrs.biotech. Today we are going to learn in detail about Introduction to Biotechnology. Through this video, I am sharing with you my self-prepared notes, very easy to understand. For complete understanding, watch full video and subscribe to our channel. Let's begin with Introduction. The term biotechnology was coined by Hungarian scientist Karl Ehricke. In biotechnology, bio means biological systems or living cells, whereas technology means methods, systems, and devices used to make products. Thus, biotechnology refers to different technologies that make use of living cells or biological systems to generate useful products. Now, let us understand historical perspectives. There are three historical perspectives: ancient biotechnology, classical biotechnology, and modern biotechnology. Now we'll see ancient biotechnology. Ancient biotechnology gives us early knowledge of food, shelter, and domestication. 10,000 years ago, in the Paleolithic era is Old Stone Age. Farmers began to cultivate crops such as wheat and barley. People collected seeds of wild plants for cultivation and domesticated some species of wild animals, which is now known as selective breeding. Classical biotechnology. In classical biotechnology, we get information about fermentation-based food production and medicine. Fermentation technology was used for production of bread, cheese, wine, beer, curd, that is dahi, idli, and kinema. Kinema is fermented soybean food prepared by Nepali community. And one more thing I would like to mention here is that fermentation is microbial process in which enzymatically controlled conversion of organic compounds occur. So, fermented dough was discovered accidentally when dough was not baked immediately. It underwent fermentation by yeast such as Saccharomyces winlocki. Baker's yeast was discovered by Romans. Around 2000 BC, that is before Christ, Egyptians started vinegar production by preserving crushed dates for longer time. Vinegar is a product which we use in cooking and also in cleaning. One more thing is that, and after 4000 BC, Chinese used fermentation technology for production of soy sauce and fermented vegetables. Around 5000 to 6000 BC, beer making began using cereal grains such as sorghum, corn, rice, millet, and wheat. East and Europe knew the art of preserving animal foods by drying, smoking, and pickling in the brine. Louis Pasteur established that yeast and other microbes were responsible for fermentation. Industrial fermentation was established during World War I as glycerol was used for explosives. World War II was the catalyst which led to the invention of bioreactor for mass production of antibiotic penicillin. The bioreactor is also known as modern fermenter. Next is modern biotechnology. In modern biotechnology, it deals with genetic engineering and recombinant DNA technology. So, genetics and principles of heredity was developed by an Austrian monk named Gregor Mendel. rDNA technology has revolutionized biotechnology by allowing scientists to cut and join different pieces of DNA and place the new recombinant DNA into new host. Recombinant DNA is also known as chimeric or hybrid DNA. Now we are going to see areas covered under biotechnology. So there are 4 areas covered under biotechnology: blue biotechnology, green biotechnology, red biotechnology, and white biotechnology. So, as the color indicates, blue biotechnology is for marine and freshwater organisms, which are used for increasing seafood supply, regulation of reproduction of dangerous waterborne organisms, and developing new drugs. Green biotechnology is for environment-friendly solutions, such as in plants to improve nutritional quality, quantity, and production of eco-friendly transgenic plants with improved traits. Red biotechnology for manufacture of pharmaceutical products such as insulin, enzymes, antibiotics, and vaccines. White biotechnology is to improve industrial processes. The use of enzymes as industrial catalysts in eco-friendly manner for the production of valuable chemicals. Now we will see industrial scenario and academic prospects in Indian biotechnology. Industrial scenario. Indian biotechnology industry is one of the fastest growing industries of the country. Presently, India is among top 12 biotechnology powers in the world and third biggest industry in Asia Pacific in terms of infrastructure. That means India has good infrastructure of biotechnology industries. The Indian biotechnology industry is divided into 5 segments, namely biopharma, bioservices, bioagri, bioindustry, and bioinformatics. Biopharma shares revenue of 64%. Bioservices share revenue of 18% in business. Bioagriculture shares revenue of 14%. Bioindustry shares revenue of 3%. And bioinformatics shares revenue of 1% towards Indian economy. India has fourth largest area, that is 11.57 million hectare covered by genetically modified crops, and the area is highly dominated by BT cotton. India is world's largest producer of recombinant hepatitis B vaccine. Now we will see the academic prospects of Indian biotechnology. NBTB, that is the National Biotechnology Board, was constituted by Government of India in 1982, which was upgraded to Department of Biotechnology, that is DBT, in 1986. DBT has established major research institutions all over India. DBT also supports postgraduate academic programs of biotechnology in the subject areas of agricultural biotechnology, marine biotechnology, neuroscience, industrial biotechnology, environmental biotechnology, bioresources, etc. Now we will see applications of modern biotechnology in our day-to-day life. These are covered under 4 categories: medicine and healthcare, crop production and agriculture, food processing, and environment protection. Medicine and healthcare cover disease diagnosis. Disease diagnosis is nothing but to identify disease from its symptoms. Next is production of vaccines. Then comes gene therapy. Gene therapy is treatment of diseases caused by gene defects. Then is genetic testing. Genetic testing helps to identify genetic composition defects of an individual. And the last is production of therapeutic molecule. Therapeutic molecules are antibiotic, hormones, proteins, etc. Another applications of modern biotechnology is crop production and agriculture. This covers crop improvement. Crop improvement helps in development of transgenic plants exhibiting resistance to pathogen, salt, cold, herbicide, etc. 5 major traits used for crop improvement are insect resistance, herbicide resistance, virus resistance, delayed fruit ripening, and nutritional enhancement. Then comes biofuels. Biofuels are the biomass that can be converted to convenient energy containing substance. Examples of biofuels are biobutanol, bioethers, biogas, etc. Another application is in food processing. Biotechnology tools help in improving the edibility, texture, and storage of food, and also has many more aspects. The last is environment protection. Biomarkers. Biomarkers are defined as any naturally occurring molecule which may indicate specific biological process in response to any environmental or chemical stimuli. So these biomarkers are present in environment, and the process of cleaning up the hazardous substance in the environment by converting them into non-toxic or less toxic compounds is known as bioremediation. So hope you all guys have understood what I have explained, and for more such informative videos, stay tuned to our channels. Bye bye, see you.